Hello, good morning to Dr. Afnani. Today we are going to talk about how CO2 affects cell wall metabolism. Our group consists of 5 members, which are me, Kang, Muhammad Ishaq Mukris, Nurul Iswani, Siti Zulaika, and Alia as well. First of all, what is abiotic stress? Abiotic stress is actually a physical or chemical damage due to the plant and can cause temporary and permanent damage. Plant cell wall actually composed of cellulose, hemicellulose, pectin, and glycoprotein and is maintained by cell homeostasis. So in this video, we are going to talk about how stress factor affect the cell metabolism and component of the cell wall. First of all, me, Kang, are going to talk about how CO2 affects cell wall metabolism. The increases in photosynthesis rate, and cell wall formation actually depends on the interlinked cellulose polysaccharides matrices such as xyloglucans. From the overview of cell wall metabolism actually consists of morphologic, transcriptome, proteomic, and biochemical. First of all, from the morphogenic subview will cause the cell wall length to extend and expand in paradermal plane. Besides that, it also modified mesophyll anatomy. All of these are due to the increasing of XTU. After that, let's go to transcriptome. Increases of CO2 concentration actually inhibit the gene expression in relation to photosynthesis and sugar metabolic pathway. This contribution will bring down the regulation of photosynthesis pathway due to the compilation of carbohydrate in proteomic level, causes the GPI to increase, the cell wall formation increases as well. And HRGP actually incorporate into the matrix cell, which provide further structural support to the plant cell wall. After that, let's go to biochemical. Biochemical induce accumulation of starch grain within chloroplasts, leading the distortion of thylakoids. That is why increasing of biosynthesis of pectin and hemicellulose to adapt to the high carbon dioxide condition, which able to limit the barrier to control the permeability towards carbon dioxide into the palisade mesophyll. Thank you. Next, I'll pass them on to Mukris, which is the next presenter.
Thank you, Kang, for the explanation. So now I'm going to continue explaining how the stress factor, to be exact carbon dioxide, affect the cell wall metabolism. So the main concept here is that primary cell wall is acting as the limiting barrier that controls permeability of carbon dioxide before entering the palisade layer or the cells. So the next one is the hemicellulose content in the cell wall of mesophyll cells increase under high condition of carbon dioxide. The example is a research on plant Betula pendula species or silver birch plant. Furthermore, cell wall elasticity also increase along with high amount of carbon dioxide. It is stated that barley plant cell wall elasticity increase in high carbon dioxide level. Moving on, the pectin-related enzyme and cell wall homeostasis such as pectin, polysaccharides, and hemicellulose biosynthesis also increase in high condition of carbon dioxide. So the last one, as the carbon dioxide increase, the sucrosynthase that are important in synthesis of both cell wall polysaccharide also increase.
The rising of carbon dioxide concentration in the atmosphere will increase the photosynthesis rate. Plants utilize carbon dioxide from the atmosphere by photosynthesis to transform it into organic chemical compounds which are needed for growth and development. From here, the increase of carbon dioxide in the atmosphere increase the carbon content in the plant. More carbon in the plant promotes cell wall formation. This causes the cell wall in plant to be thicker compared to regular plant cells. The organic chemical compounds are constructed by carbon, where carbon is essential for cell wall formation.
In order to understand the complex interaction between expanding cells, we need a profound understanding of the mechanical processes during growth at the single cell level. The mechanical properties of the plant cell wall are defined by its biochemical composition and the specific interactions between individual wall polymers. It is depending on the formation of interlinked cellulose and polysaccharide matrices. Cellulose present in the form of cellulose microfibres which synthesize at rosette terminal complexes. Matrix polysaccharides are synthesized in the Golgi, then exported to the cell wall by exocytosis, and then intercalate among cellulose microfibrils.
Hi, I am going to present regarding the application of the carbon dioxide. How can we apply the carbon dioxide in life? So, we can increase the yield by elevating the carbon dioxide surrounding the planting environment. Meaning, we can use the greenhouse that equipped with carbon enrichment for a plant stimulation system that also utilize the land usage more efficiently. First, we can use the compressed carbon dioxide tanks, which compress the liquid form of carbon dioxide, which then vaporizes to the vaporizer and diffuse throughout its distribution system. The second one is the carbon dioxide generator, which produces the carbon dioxide, water, and heat through the hydrocarbon fuels combustion process. It also needs oxygen in the proper amount to prevent impurities produced. So in the overall conclusion, the increasing amounts of the CO2 in the environment now is forcing plants to adapt a new way to survive in this current situation. The mechanism and the composition of the cell walls affected by the abiotic stress, like for example this elevated carbon dioxide. It is different from one species of plant to another, also because of the responses to climate change are actually species dependent.